My presentation is about the application of conotoxins as molecular probes, therapeutic leads, and drug delivery vectors in excitable and unexcitable systems. My name is Alberto Padilla. And my story today is about these interesting marine gastropods. These invertebrates are venomous and use their venom to capture prey and deter predators. Their venom is a cocktail of thousands of compounds with immense therapeutic potential. Venomous marine cone snails range from 2 to 10 centimeters in size and are distributed along the Pacific, Atlantic, and Indo-Pacific Oceans. In our lab, we keep our healthy cone snail specimens in aquaria and individually labeled. Having introduced you to these prolific molecular engineers on Earth, let me now tell you what my research is about. It is to discover novel molecules with therapeutic potential to treat diseases related to the nervous and immune systems, and to synthetically mimic their chemical structure and biological function. This image is a schematic representation of the receptors, ion channels, and transport molecules present on the surface of cell membranes and are target to components in snail venom. It is here where we test our purified compounds from the venom. Blocking these cell surface proteins result in physiological disruptions. What you see in front of you is a flowchart of the experimental approach of our research after having collected the specimens. The first phase is entirely wet chemistry: extraction, isolation, and purification of molecules. The second phase deals with testing the bioactivity of the molecules purified from the cone snail venom in excitable and non-excitable systems. These are pictures of the laboratories where we do our wet chemistry, mass spectrometry, and nuclear magnetic resonance. To collect the venom from some species of cone snails, the snails are enticed with a piece of fish fin affixed to a piece of parafilm bound to a microtube. The snail sniffs and strikes depositing the venom in the tube. Left image. Immediately after, we feed them with a live goldfish. Right image. Other species are not so lucky, and we have to dissect the venom duct from them in order to obtain the venom. In order to extract microgram to gram yields of peptide, we dissect close to 500 specimens. The venom ducts are then placed in a 50 ml polypropylene conical tube, homogenized, and centrifuged at 11,000 RPMs at 4 degrees Celsius. After 3 rounds of centrifugation, as seen on the right-hand picture, we end up with a very clear supernatant containing the molecules of interest. The next step is to lyophilize the supernatant containing the molecules of interest in a vacuum at -80 degrees Celsius. The compounds are stored in dry form at -80 degrees Celsius or used in further research. The lyophilized venom powder has thousands of molecules in it, so we first separate these molecules by size using size exclusion chromatography. We choose the most abundant low molecular mass peak represented above as peak number 6, and further separate molecules according to polarity by reverse phase high pressure liquid chromatography at bottom. Each peak in the reverse phase liquid chromatography spectrum represents the presence of a purified compound. Each one of the peaks representing molecules in the reverse phase high pressure liquid chromatography that you saw in the previous slide are collected in individual plastic tubes. These tubes are stored at -80 degrees Celsius until we test their bioactivity. We next determine the molecular mass for each purified molecule by mass spectrometry, seen on top, and quantify the amounts by ultraviolet spectrophotometry at 205 nanometer wavelength at bottom. We further purify molecules until a single peak is seen in the mass spectrum and verify this by nuclear magnetic resonance. In the next step, after molecule purification, we test our peptides for biological activity. Here, specifically, we determine activity in human voltage-gated sodium channels that are present in the central nervous system. The channels are expressed in all sides of xenopus levis. Our results indicated that one purified peptide was able to block the human voltage-gated sodium channel 1.6 with over 52% efficiency. This is quite good for a natural compound. From this lead, a synthetic form can be constructed chemically to optimize activity. Next, using proteogenomics analysis, we characterize the biologically active peptide using tandem mass spectrometry and NMR and construct the three-dimensional molecule observed on the left. Results will be hopefully published in a peer-reviewed journal next year. As we continue to move along our experimental flowchart, we developed assays to test other purified molecules in non-excitable cells. One such assay is a high-throughput bioplex assay to determine the capacity for cone snail venom components to block the host-parasite interactions in the microvasculature and the placenta that occur during malaria infection. Host-parasite interactions can be seen on the left and the mechanism of infection on the right. This image represents how our high-throughput bioplex assay works. Let's focus first on the top part of the slide. It makes use of green fluorescent protein antibodies that are fixed onto polypropylene beads to capture Plasmodium falciparum protein domains. A biotilynated receptor will bind to the protein domain if no interference is present. Next, a streptavidin conjugated to phycoerythrin binds to the biotilynated receptor and emits fluorescence, as seen on the left-hand side of the fork. If a purified peptide binds either the domain or the receptor of this reaction, as seen on the right-hand side of the fork, fluorescence is not produced, and the results are interpreted as inhibitory. From the bioplex assays, we learned that several Plasmodium falciparum host interactions can be disrupted with purified cone snail molecules from the venom. The smaller the bar with regard to the corresponding negative control bar in each one of the graphs at left indicates the levels of inhibition by these molecules. Many conopeptides inhibited equally or better even than the positive control. With such motivating results, we then turn to proteogenomics and computational biology using tandem mass spectrometry, de novo sequencing, and 3D NMR to construct the image of the natural compound and synthesize it chemically. This work was sent for peer review publication to the Journal of Proteomics on October 9, 2020. In our research, we also developed a cell-based immunoassay to understand how the central nervous system regulates the immune system. Using the synthetic form of the conotoxin seen at center top, we found that the central nervous system does not regulate the nervous system directly through the vagus nerve as depicted on the left image. Instead, we found that vagus nerve innervation to the spleen releases noradrenaline that targets the beta-2 adrenergic receptor on the surface of resident helper T cells, which in turn expresses acetylcholine to downregulate the immune response via the alpha-7 nicotinic acetylcholine receptor present on the surface of macrophages. Our results on the regulation of the cholinergic anti-inflammatory pathway using leukemic cells as model was published in a major scientific journal. In summary, conotoxins are highly diverse molecules that target cell surface proteins with high affinity and specificity, hence making them remarkable therapeutic leads applicable to neuronal and non-neuronal systems. I would like to recognize the people who were involved in one or more stages of my research: Dr. Frank Marie, Dr. James Hartman, Dr. Andrew Olenikoff, Dr. Patricia Keating, Dr. Olga Shesnikoff, and Dr. Mikaeline Hoggard.